Good afternoon, everybody. I'm so pleased that you could join us. My name's Tom Chandler. I'm Dean of the Arnold School. I think most of the folks in the room are Arnold Schoolites, but welcome all those from around campus that came out for this seminar as well today. This is our, it's counting up, I think this is our 7th annual Delta Omega Honor Society and Public Health Lecture, and we took a charge several years ago to try to recruit speakers that have a strong appreciation for the discipline of public health, speakers that could convey the importance of public health as a discipline, but as importantly, convey the importance of public health in the community. Our speaker today, who you'll hear more about in a moment, was recruited by our distinguished professor of exercise science, Steve Blair. Steve came to my office back in, I believe, November and said, Tommy, I know this guy out at Stanford, and he's got more than 1 million hits on a particular PLOS One paper, and Steve thought he might be of interest to the community. Of course, I'm a dean now, and I don't keep up with literature well enough, and frankly, John, I had not heard of you before, but I went online, and then I was really impressed. So such is the life of administration. But anyway, welcome you. Steve, if you'd come up and just give a few remarks, and thank you all again for coming out, and let's have fun today with John. Show him our hospitality, but also put him through the ringer a little bit. I think it might be fun. So.
No, he can handle tough questions, I assure you of that. It is a pleasure to see you all here, and I am making a wild assumption. Can everybody in here read? You can read? Well, then there's all this information about John on this sheet. So it really irritates me when I go somewhere to give a talk and somebody reads what's written there forever. So I'm not going to do that, John. I'm going to think of something else to say. But John certainly is a distinguished scientist. I say he's one of the great scientific minds of our generation. And really so pleased, John, that you decided to come here and spend a little time with us and give a lecture. And some of you know I'm a little crazy about H-index and Web of Science and citations and so forth. So I do want to tell you that, John, it's really disappointing. You only have 5 papers cited more than 1,000 times. And the most recent of these was published in 2009. 1,000 papers, or 1,000 citations of a paper published in 2000. That's a tough, you're going to have to struggle to beat that, I'm afraid. And 8 papers cited more than 500 times. So John is clearly a giant in several areas of science. You're going to really enjoy his lecture. So John, please take the podium.
Thank you for the very kind invitation. It's really a great pleasure and an honor for me to be here. And I have already enjoyed the hospitality that the university has offered me. And Steve in particular, including spending a night at his home. So there's some tension in my title. There's objective, but there's also quality. And quality is subjective. So when we're trying to evaluate research, the question is, can we do that objectively? And even a step backwards, why do we want to do that? Just to get promoted, for example, or to give funding to people, or maybe better to get reliable results, to get something that is credible, that we can build on, that we can take the next step. So I'll try to venture in some of these ideas. How many scientists are out there? Probably about 15 million scientists have published in 1996 to 2011 in Scopus. That database, which does not include everything, just a limited time period and not all journals, just 13,000 journals out of about 100,000 journals. It includes 25 million papers plus in that time frame, and recently an estimate is that we have 120 million scholarly documents across all sciences and all times. So clearly a plethora and an embarrassment of riches. And each empirical paper can include anywhere from a few up to many millions of results. How many scientists? So I told you 15 million scientists, authors just in Scopus. But if you look carefully in an analysis that we published about a year ago, we found that only 1% of those scientists have authored at least one paper each and every year in the period 1996 to 2011. So this is like the continuous and uninterrupted presence core. And that core is very influential. It accounts for 41.7% of all papers in that corpus and for 87.1% of papers with over 1,000 citations in the same period. That core is getting bigger. So about 10,000 scientists net gain every year, which is a composite of some people stopping for various reasons, retiring, dying, quitting science, and others becoming uninterrupted present scientists for so many years. But still, the core is pretty small compared to the very large community that science has engaged nowadays. How can we assess the research potential and impact of all these sciences, scientists and papers? So maybe we should look at significance, maybe we should look at innovation, maybe we should look at whether research is rigorous and reproducible, or try to judge investigators or also teams or departments or institutions in terms of merit instead of just trying to do that for one analysis and one paper at a time. So how easy is that? Significance is not easy because almost all scientific papers claim that they have found statistically or conceptually significant results. And obviously, all grant proposals claim that what they do is highly significant. I don't think that I ever submitted a grant saying that I'm not going to find anything and my research is unimportant and this is why I want to be funded. Along with David Chevalerias, we looked across PubMed and we found 1.5 million abstracts with p-values, and 96% of them report statistically significant results. Actually, do you think that scientific fields that report more significant results are more reliable than others? So Dan Fanelli, who recently joined Metrics at Stanford, believes that it's the opposite, that results that are too significant are just too good to be true. And if you look at space science, it has less significant results compared to other fields that have almost everything being significant. And almost everything being significant includes things like lots of medicine, epidemiology, but also psychiatry, psychology, pharmacology, and economics. Innovation. How can we judge innovation? There's hardly any reliable method to predict innovation ahead of time. I may make a claim that my research is innovative, but how do I know? Did it really change the world? How much do I have to wait to see that it does change the world? Usually, it takes many years, often decades, to materialize and translate research into something that is eventually innovative. And probably the vast majority of scientists who spend their very best efforts never manage to create something that is highly innovative and really changing the world, despite the very best intentions. This is a survey that I published last year in Nature with colleagues from the Map of Science, asking the most highly cited authors across biomedicine to tell us what they think of their most highly cited papers published in 2005 to 2008. So not too long ago because things get stereotyped, and not too soon because obviously it's not easy to accrue tons of citations. One would think that these are extremely influential papers and innovative, but actually the main contribution of these papers is continuous progress, broad interest, greater synthesis, so incremental steps, one step at a time, rather than disruptive innovativeness or surprise, something that came out of the blue and nobody had thought about that. And obviously these are the most highly cited people, so the publication difficulty score was not very high. But again, these are the most famous and influential scientists, and also these are their most highly cited papers. So I think that it's expected that we should see that. Innovation and incremental progress are not opposite forces. This is an analysis showing the correlations between these 6 variables in that cohort of highly influential papers. And you see red links, which is positive correlations. There's no blue link, which would be a negative correlation. So disruptive innovativeness, for example, does not correlate with continuous progress. But they're not opposite. They're not negatively correlated. They're complementary. So we need both. We need good science that would be the major discovery that really opens a new avenue, but which is not very common. And we need good science that will continue adding some incremental knowledge, information, and eventually applied interventions. This is another empirical evaluation that I did with my team a number of years ago. We took 6 journals that are very prominent in what people quote as basic science. I would say bench science may be more appropriate for the description. So Science, Nature, Cell, Journal of Experimental Medicine, Journal of Clinical Investigation, and Journal of Biological Chemistry. And we tried to find discoveries that made clear promises for eventual clinical applications. So the paper said this will have a major impact in clinical medicine and or public health. So out of 25,000 papers, we found 100 that had such a clear-cut promise. And how many of those do you think eventually worked? Only 1 in 4 resulted in a published RCT. Only 1 in 5 were validated in at least one positive trial. Fewer than 1 in 10 entered routine clinical use within 20 years of the quote-unquote basic science promise. Only 1 has been used extensively for the licensed indication. And even most of those that had quote-unquote positive trials with significant results, eventually they were not adopted into clinical practice. So this is the creme de la creme of the best bench science discovery-oriented journals, the papers that made the most prominent promises, and yet after 20 years, and we have updated these results now with almost 30 years, picture is not different. You know, just one application that made a huge impact, ACE inhibitors was one of these promises, and another 4 or 5 that were licensed, but they're not really mainstream first-line treatments. This is another look of the same phenomenon of delayed innovation. This is the most highly cited papers in biomedical research with clinical applications, plotting their history. So if you see a red point, that's when a clinical trial was published or an epidemiological study was published that had been cited more than 1,000 times afterwards and established that potential intervention. And you see some black, which means that about 40 of them, even though they were supported by such extremely highly cited papers, eventually got refuted by even better, larger, better controlled studies. And going back, you see the other milestones when you had the first description of the intervention class, the first description of the intervention. I hope you can see the, oh, you cannot see anything. I can see the pointer myself, but, so Steve promised that he will point to whatever I say, but he doesn't know my talk. Oh, okay. Forget it. I will try to explain. So the different colors are showing the first description of the intervention, the first description of the very specific intervention, the first article about human use, the first article of the specific setting human use, and then you have the red color, which is the highly cited paper, and occasionally, actually pretty frequently, the refutation. So on average, we're talking about 20 to 30 years, 25 years on average from the discovery until you have that major hit. And even for these major hits, about 40% of them seem to die with better evidence. So one option is to wait 30 years before we decide on whether we will promote someone. to make sure that it's a good investment. But probably this is not a good solution. So one possibility is to forget about single papers and focus at the big picture of the contribution of someone to science and public health and the literature, and try to understand, well, are we dealing with a good scientist, with someone who has a track record that would be good enough? Forget about the very specific project here. Maybe we cannot really judge that project because there's lots of anticipated but also unanticipated consequences. But if we're dealing with a good scientist, we should fund her. And that would be the way to move forward. Now, if we were to do that, then how is that going to happen? There's many possibilities. One is that, oh, this is too difficult. Let's just give some money to everybody. So we take all the funds that we have and we split them in an egalitarian mode. Or we could do it at random in an aleatoric fashion. So 20% will get funded, kind of winning the lottery. Or we could try to assess career trajectory and this is what people do most of the time. They get a CV and they read, that I climbed Mount Kilimanjaro when I was at high school, and then I was a top athlete at Stanford, and now I want $20 million for my research, whatever. This is very subjective. So maybe we can use automated impact indices, as Steve suggested. So we can decide what these indices would be, and we can try to test their construct validity, face validity, how they correlate, what they mean, what their predictive ability is for future accomplishments. We need to add scientific citizenship probably to this, depending on what we want. Eventually, do we want a monster who's closing the door of his lab and is working day and night and cannot communicate with anyone, does not contribute anything to the life of the institution, or we want someone who's also offering in different ways? And then how are we going to measure these ways? I mean, just number of committees that someone is participating. This may be probably a negative component of appraisal. I mean, how do you judge citizenship is not easy, but maybe we could have some quantitative features about that. Or we could try to merge a piece of person being appraised versus end project, but project with very broad goals. That I have these very broad goals. I'm working in exercise physiology, and there's a couple of things that I try to do. Just leave me alone, and I'll try to do my best to solve these problems. Who will be the judge, and based on what? Peers. Sounds great. People who know and who are in the same domain. But here we have some problems. There's, for example, evidence, and actually much of that evidence comes from people not in biomedicine but in behavioral economics and sociology, that peer review is biased against innovative, high-risk, out-of-the-box ideas. And even more so in federal settings when there is a fixed budget and people feel that they have to justify that I didn't invest in something that would be weird, but I will get my money back. That bias can become even more impressive. Then people who conform are more likely to compromise and participate in a conformity-based system. So they may be able to engage themselves better in a community where there is some quid pro quo, in a sense, and some engagement with other colleagues that respect you, and you will support them, they will support you. In a vicious circle situation, the system may eventually select people who are more alike to those who are already insiders, and then you just get more of the same. You don't get something that is new, novel, unusual, a new avenue. You just continue doing what you were doing 10 or 20 years ago. So I tried to see whether that rationale makes sense and whether it is happening in an empirical analysis that we published a couple of years ago in Nature. So we said, let's take people who have published papers as first authors or last authors with over 1,000 citations in the literature since 2001. And there's not that many people. So there were a little bit over 1,000 all over the country and all over all types of biomedical research, very broadly defined. And we found that 94% of them are not serving in NIH study panels. So first observation is that these people probably don't have time to do that. So the very best scientists very rarely participate in that sort of peer review. Second observation is that 60% of those people who did not have NIH funding. So they had produced the most influential work in the literature with the major repercussions, but they didn't have any grant currently. And we tried to probe why was that happening, and we've realized that many of those had even stopped being engaged in academic research. You know, they had become very successful as entrepreneurs and still doing influential research, but they just didn't tolerate the academic system. So in a way, we lost some of the best minds to a competitor, and maybe it's not a competitor because we need both. We need both academia and industry and entrepreneurship and everything. But to me, at least being on the academic side, I think that it would be nice to have these people next door. And for all of us, I think it would have been nice. So reviewers fund projects that are similar to their own work. Based on the NIH reporter data that we looked at, grants of study section members were more similar to the other NIH-funded grants than it was the case for grants of non-study section members. So you can estimate a similarity score based on what are the concepts being described. It's practically a text mining exercise, so it's not very sensitive, but it can tell you very quickly whether a grant has some similarity and familiarity with other grants in the same area. Here's some data on the study section members. The vast majority of NIH study section members have never published a highly influential paper themselves. Their citation credentials were not bad, but they were not excellent. And you know, as I mentioned up front, most of the most influential scientists, they don't do that for whatever reason. This is showing the percentage of study section members that have a paper with X number of citations as being their most highly cited in their career. And the vast majority have had their best paper have about 100 citations, more or less. This is how it looks like, median of 136 citations, interquartile range of 90 to 229. Total citations less than 2,000, H-index median of 20, and only 2% with over 10,000 citations. So decent, good, but the very best are not participating. Well, how about academic technocrats who are really running the show and deciding on what direction science would take? So this is an analysis that I didn't publish because I didn't want to get life threats, but I looked across different countries and tried to identify ministers, in some cases even prime ministers, who were professors at universities and researchers. And their age index for the entire career was 4, which is probably much less than many of our graduate students. And only 14% had an age index exceeding 18, and only 27% had authored at least one paper that had received over 100 citations. So, the people who run the show, even when they come from an academic life, and not just running the show at an academic level, but at the level of political decisions that affect the society at large, somehow the best people are even more disencouraged to be part of that game and get these positions. So then we have another confounding factor, which is the fact that science has been so successful and we see so many papers published has led to what people describe as a reproducibility crisis. It means that empirical studies across different domains suggest that most of the claimed statistically significant effects in traditional biomedical research are either false positives or substantially exaggerated. I will spend a little time on that because I think the two phenomena are very tightly related. I mean, the fact that we get so many papers, and so many papers are not really replicable, it means that we have a system under a lot of pressure. So if we could solve one problem, maybe we could solve the other. So here's some evidence. This is one field where replication research became very easy and very cheap, genetic epidemiology. You know, the cost went down 100 million-fold over 10 years. Now we have very big consortia with genome-wide association studies and sequencing. So we could test whether the old proposed associations of genes associated with disease hold true now that we can test them robustly in large-scale evidence. The replication rate is 1.2%. So 98.8% of the original claims published in the very best journals could not be replicated downstream. And most other fields don't really have gone through that transition where the cost of conducting research and experiments has gone down 100 million-fold. So we don't have that much replication happening, but whenever we try, we see that it just doesn't work. This is another field. This is looking at the most highly cited papers on biomarkers in the biomedical literature, most cited of all times. And you have on the horizontal axis the relative risk in the highly cited study, and on the vertical axis the relative risk in the largest study. And you see almost all the actions below the diagonal, which means that the highly cited study made a big claim about a very strong effect, and then when we performed a large study, that effect went away. Now, it didn't go away completely. If you look at these points, they're not relative risks of 1. They're 1.2, 1.5, so there is still something there surviving, but it's not a big effect. It's much smaller, and for practical terms, from a public view perspective, much of that biomarker machinery that is actually at the foundation of the claims for personalized medicine is not good enough to be able to be used in real life. I mean, if you have a biomarker with a small relative risk and modest discriminating ability, it's good to know for scientific curiosity, but it's not something that I would like to screen the population or even specific subpopulations for practical purposes. A pleasant surprise is that in the last few years, the industry has been championing replication. That's kind of weird. You know, for many years, I have been talking again and again about the industry needs to be transparent and reproducible and avoid conflicts. So the industry has shown interest in replication because they don't want to waste money in developing drug targets that will not work. So they're using preclinical research, and there is a number of series trying to reproduce preclinical research on drug targets. 75 to 90% of the most highly cited papers on drug targets published in the very top journals, Nature, Science, Nature Medicine, and so forth, are not replicable. Even when you ask the investigators and you try to get the exact recipe of how did you do that, it cannot be done. This is Glenn Begley and his team finding that only 6 of the 53 landmark studies for Amgen oncology drug target projects could be replicated, and concluding that the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. Some countries are doing even worse. So here's an analysis that I did with Dan Fanelli, trying to see whether there's an over-exaggeration of effect sizes when you take behavioral sciences like psychology and psychiatry, and genetics, which with all its caveats still has very accurate measurement. You know, genotyping error is very tiny. Well, in a psychological measurement, you can have some flexibility. We found that US-based studies tend to overestimate effect when you have subjective measurements. Now, it doesn't mean that they don't suffer or suffer from more publication bias than studies in other countries. Publication bias is pretty much the same, probably. But there is some over-exaggeration for what gets presented. The effect sizes are even bigger, more extreme in both directions when they come from the US. And one is wondering whether this is a reflection of the fact that maybe there's more pressure. It's a more competitive environment. I think we have better scientists on average. We have more competitive scientists. We have a shrinking budget or stable budget with an expanding pool, more pressure. So more pressure to deliver exceptional, extraordinary, extreme inflated results. Why does this happen? Practically, this happens even with the best intentions and even to the best scientists. And the reason for this is that there's practically two forces that create non-credible results. There's bias, or actually many biases, and there's random error, which you can see also through the window of multiple comparisons. Lots of things being analyzed by many people. Usually there's plenty of both. And almost no study really takes into account the total multiplicity of what's happening in the analytical space. I mean, many people may attack the same database, they may attack it in many different ways. Each time we see either unadjusted p-values, uncorrected, or they may be corrected and adjusted for the sample of analysis that were done within that particular database. As far as bias is concerned, along with David Chevalier-Rias, we looked at 17 million PubMed papers with text mining, and we mapped 235 biases. I'm considered to be an expert on bias of sorts, but I have to say that many of these biases, probably most of them, I had never heard of before I found out about them. So how many of the studies that I have designed, I have carefully anticipated and corrected and taken care of all 235 biases? None. I mean, usually I think of a few that are more prominent, but there's still plenty of them out there. So we have discovery, and we want to reward the best discoveries, and we have replication, and we want to reward the best, more rigorous replications. And then we have the pairing of the two. And we can have these different scenarios where discovery can be correct or wrong, and replication can be correct or wrong. So how common are these scenarios? We have some evidence from some fields. This is putting together empirical evidence from about 12 empirical investigations that were published in late 2012 in Perspectives in Psychological Science, and I was asked to try to put together their conclusions. So the optimal scenario is that the discovery is correct and the replication is correct. And apparently this is happening in psychological science less than 1%. Then discovery could be wrong and replication is correct, so we correct our error. This is the self-correcting paradigm. Apparently, this is also occurring less than 1%. The discovery may be correct, and the replication may be wrong. It's possible that the replication study has some flaws or whatever other reason causes a result that is not what we should get. This is also less than 1% likely to happen. Perpetuated fallacy, which means that the discovery was wrong, the replication didn't correct the flaws or biases that were embedded in the replication, in the original discovery effort. So we got the same false result. This is about 2%. Unconfirmed genuine discovery, which means that we perform some study, we find something, but nobody cares to replicate, or there's no funding to replicate, or I'm not going to do Me Too research, 43%. And unchallenged fallacy, we got something wrong, but nobody is challenging it, 53%. It stays in the literature, but that's it. So I'm not saying that this is typical of all fields, and I have to acknowledge that there's some error bar around all of these estimates. But if this is really the picture that we're dealing with in even a few scientific disciplines, I think it's really problematic. And there's many empirical studies now from very diverse disciplines that have shown reproducibility concerns, usually of that magnitude or even bigger. So we have seen problems with reproducibility in microarray studies, in animal studies, in general biology, in oncology studies, in genome sequencing data, in vivo mouse studies, neurological models, mouse models, genomic cell line, commercial antibodies, ELISA-based studies, and also behavior of journals surrounding reproducibility practices. And this is a number of fields that have witnessed problems in preclinical basic bench research, neuroscience, pharmacology, genomics, bioinformatics, stem cell biology, oncology, in vitro or chemistry-led discovery, computational biology, pathology and biomarkers, organizational psychology, and observational research in general. There have been empirical studies that describe similar problems to what I presented to you, obviously each time a little bit different. Big data is another factor that we need to take into account as we move forward, because as I said up front, we're having an increasing ability to measure and analyze things. And most people see big data as a blessing because we have more things to analyze. And this is very nice, and I can spend more time analyzing all these data sets. But at the same time, this creates also the possibility for big noise and for big error. I wrote that paper with Moin Khoury a few months ago for Science, where maybe we express a bit of a heretic view about big data. But I was surprised to find that there are a number of sympathizers in the community. And lots of people felt that, indeed, it's a great opportunity. But if you see it from the perspective of the false positive rate, it's a wonderful opportunity to escalate the false positive rate. Then we have what I call stealth research, which is research happening outside of the peer-reviewed literature. And many innovators believe that they should have nothing to do with the peer-reviewed literature. You know, at some point they felt that for whatever reason that system doesn't work, and I'm just going to create something that's important. I will probably file a patent to secure my monetary compensation downstream, but why should I publish that? I don't have time to waste. And unfortunately, this is becoming very frequent. There's lots of big claims about technology that would change healthcare and our lives and disrupt the medical system and offer new opportunities, not necessarily on drugs, because obviously drugs need to have some registration of trials and some pressure to get some results published, but lots of other technologies, they're a little bit out of the publication targeting. And how much can we trust those if there's no peer-reviewed literature? I mean, sometimes it's very obvious that something works. An airplane flies and you can get from here to New York or to Stockholm. If it crashes, then you know it doesn't work. But how much of medical research or public health research we have such clarity? Oh, it crashed and everybody, burned and died. It's not so easy. I mean, we may see some big trends and some big changes in before, after, but as you all know, these are very precarious conclusions. So some possible solutions to improve some of the current research practices is what I'm going to spend the next 10 minutes and hopefully leave some time for questions and discussion. There are some key features about the solutions that are being proposed. And I see lots of solutions being proposed all the time in the last few years. Somehow the whole theme has become very popular. So solutions may work best when they're integral to the reward and incentive system. If we want to improve something, I believe we need to integrate it with our reward and incentive system and with how we do research per se, rather than just try to create yet another bubble that would be disconnected from our everyday life and our everyday happenings as scientists. There are some people who claim that there may be even opposite effects, even when you do that. The classic example is the literature on what happened when you try to impose fines to daycare when you're late to get your child. So fine, $50 if you're 1 hour late. You should better do your job, your scientific job here being get your child on time. Does it work? Actually, it increases the rate of parents who arrive late because it removes from them the stress that, oh, I'm a bad parent and I left my kid, and I will pay that amount and I will be fine. It's not something that I should feel ashamed about. So we have to be very careful about how we intervene in changing the system. We can easily make things worse. Some solutions have already worked, so maybe we can take these and translate them into wider settings. Others are more speculative. And I think no matter what, we need empirical evidence on whether they do work or not. It's not something for committees to decide. Much as you do not want to have interventions about public health or medicine judged by committees in the absence of evidence, I think we need evidence and not just committees to decide how to change scientific practices. So a year ago, last October actually, I published that paper in PLOS Medicine trying to repent for my 2005 paper on why most published research findings are false. I decided to write something on how to make more published research true. These are some potential recipes, and I will run through some of them. One recipe is to reward large-scale collaboration and adoption of replication culture. This is a Manhattan plot. It's a very standard feature of genetic epidemiology analysis for the last 10 years or so. And this is actually a Manhattan plot for searching for genes that regulate smoking behavior. Before we joined forces and had a very big consortium with 200,000 participants worth of data and 200 investigators joining forces, we had discovered 659 genes that regulate smoking behavior, and published in the very best journals. When we did that large-scale exercise, none of them survived. And that large-scale exercise identified 4 genes. Then we had some even bigger studies. Now we're up to 7 or 8 genes that have very rigorous support. Are we saving the world? No. But at least we know that these 7 or 8 genes have very strong support, while the 659 of them that we had before, that were published in the very best journals, that were funded by NIH grants and made lots of new professors, they were just false. Reward registration. Clinical trials, I think, have seen a major transformation with registration practice. At least we know what is out there. We know basically what is out there. We don't know exactly how that trial is going to be done, so we're still missing lots of important details about the protocol and how it's going to be implemented. But I think that it's a step forward, at least to know that when you see a study published in New England Journal of Medicine, you can go to clinicaltrials.gov and see that there's 10 other trials. So we'd better wait to see what some of the other trials show before we jump the gun and say that this is it. Or you may see that there's nothing else, and this is it, and that's the best that we have to make decisions. What do we do with other types of studies? I think that it's going to be precarious to say that we will register everything. So, and very scientists feel very uneasy about that, because much of research cannot really be registered. I have to acknowledge that there's a lot of research that is exploratory, very dynamic, very flexible, discovery-oriented. I have no clue really what I'm searching for, but I'm searching. And if you ask me to register that protocol, what protocol? I have no protocol. I'm just searching. Is that a good thing to admit? Well, if that's what I'm doing, that's what I should say. If I just try to create a protocol while I'm just going wild in the data set or just thinking about crazy possibilities, it would be unwise to say that I do have a protocol while that's not the case. So I would argue that we can have these different levels of registration. Level 0 would be no registration. But this has to be transparent. I'm working in this field in a totally exploratory fashion, and this is what I do. Obviously, whatever I find needs to be replicated, better be replicated, because otherwise the credibility is likely to be pretty low. The yield is not going to be very high, but some of very interesting ideas may materialize. Level 1 would be registration of the data set. Level 2 would be registration of the protocol. Level 3 would be registration of the analysis plan, which is separate from the protocol. Even very experienced statisticians, I have talked with many superb colleagues, they admit that the analysis plan is formulated after the data have been collected, everything has been crystallized, and to some extent, maybe sometimes this is justifiable because there may be things that have not been anticipated up front. But if we could register the analysis plan, I think, up front, that would give me some extra credence that this is not something that was generated at the last moment. Level 4 would be registration of the analysis plan and the raw data, and level 5 would be open live streaming, which means that you go and you open your computer and everything you do, anyone can see it as it's happening and they can send their comments while it is happening. How much of that is real and how much of that is just a utopia? This is an empirical evaluation that we published 3 years ago, looking at the 50 most highly cited papers, most highly cited journals, highest impact factor journals across science, and trying to see whether they have policies in place to share materials, to share data, to have open access to all their raw data, to their protocols, and also whether that was a prerequisite to publication. If you see a green color, this is very good news. It means that they do have such policies in place. And there's tons of green color, which means that many of these journals are sensitized to these issues. We repeated that exercise recently. We haven't finished it, but it seems even better. Even more journals would have green color. But the last column, which has very tiny font, and I'm not sure you can read it, there's tons of zeros, which is how many out of 10 papers from each of these journals actually adhere to these policies. So people are talking about it, but it doesn't really happen. So the question is, who is going to enforce this? The journals are trying, but probably doesn't really work. The funders are trying, but maybe they're not doing it very thoroughly. Institutions could help, if they judge their promotion and reward system based on some of these features. Who will take the time to share data? When it will be done? To avoid people saying, oh, you're stealing my ideas and my data and publishing what I should have published. How is that going to be done? Plenty of questions. A couple of years ago when we published that paper, we found 25 major questions, and most of them remain unanswered, although there are ideas about how to do it. What I would argue is, regardless of which of these ideas are the best, some of them should be tested, and some of them should be piloted, and some of that should be done to see whether we can make things better. Would we get surprised once we share data? Sometimes we might. So here's a reanalysis of randomized trials. We tried to look across all PubMed to see how many occasions we could find where people had reanalyzed randomized clinical trial data for the very same hypotheses, the same PICO as the original trial. We found 37 such occasions. 35% of the time, the authors concluded something different compared to the original paper. Different in major ways in terms of the conclusion whether people should be treated or not with that intervention, or who are the patients who should be treated. Maybe it's the enemies of the original investigators who did that to put them to shame. Actually, almost all of these refutations were by the very same team and the very same authors. Why did they kill themselves? The only explanation is that in the current environment, the only way to publish yet another paper was to say that I found something different this time. Reward better statistics and methods. I can spend hours on this, but we have run out of time. We need transparent statistical analysis plans. We need statistical training and improved literacy, numeracy of the scientific workforce, better study designs. Some features can be standard, and here's one example. This is in vivo animal research, plotting the percentage of studies who have different quality measures. Things like blinded assessment of outcomes and randomization should be the default. There's nothing difficult about it. It can be done for any study with animals that you want to test an intervention in. However, still, it's only the small minority of animal studies, animal trials, that have randomization and blinded assessment of outcomes. So institutions, I think, could help materialize some of that agenda. So you can think of an institution aligning their practices to these standards. Not adding more bureaucracy, because that's something that nobody would really enjoy, but making science easier and better, if anything. So that could happen at the level of students and postdoctoral fellows, how they're trained in methods and experimental design and statistics. It can happen at the level of investigators in terms of how they design their studies, how they're trained, how they have continuous education on methods and methodology. And it can also happen at the level of the institution and how it deals with promoting the best practices, whatever these are. then you can have other centers that could help promote the cause of good quality science. There's many initiatives that are trying to promote reproducible results, like the Center for Open Science and the Reproducibility Initiative. Metrics is the new center that we launched about a year ago at Stanford, trying to study research practices and find ways to improve them. Eventually, we may need to re-engineering our reward system. Until now, we are rewarding productivity. More papers and more grants, ideally, but this may not be enough. We need to add the full dimension of the PQRST, as I call it. So P is productivity, which I think can be recast instead of more papers, say fewer papers perhaps, but with high impact, being very prominent and influential in their field. Q would be quality. So what is the quality of the work? And each field can decide on what are the quality indicators, like blinding or randomization or whatever. R would be reproducibility. So if I publish 5 extremely highly cited papers, but they're refuted, maybe I shouldn't really get the same credit as someone who publishes 2 papers but are replicated. S would be sharing. So rewarding people because they share their data and their protocols and their materials, and they do that in a way that it enhances the scientific work. And T, translational impact. Not for early discovery science, because as I said, this is going to take 20, 30 years to happen. But for people who say, I'm an applied scientist and I'm wanting to do this in order to produce that outcome. Did it happen? So I think if we do that, most scientists would not want to be applied scientists. But I think we should be judged also on what we have achieved in terms of real life. So here's one example. When you look at a productivity index, how things can be different depending on what angle you try to measure. Number of publications has tremendous potential for gaming. As a hoax in Science showed about 2 years ago, the worst possible paper, a fake paper, was sent to 300 journals. 150 of them accepted it. Another 50 were thinking about it, and the minority rejected it. So anyone can publish as many papers as they want nowadays. That's not a big deal. Cumulative impact factor of the journals. Again, there's potential for gaming, because people will game the impact factor, and there's many ways to do that. Total count of citations, or H-index, has modest potential for gaming. It's much better. Co-authorship adjusted citation indices like Schreiber's HM or harmonic adjustments, even less potential for gaming. Combining H-index, co-authorship adjusted indices, and total citation count gets even better. The above plus excluding different types of self-citation, either by the person or by the team, even better. The above plus in-depth scrutiny of single papers in terms of their quality and what they say and whether they have been replicated, very limited. And the above plus consideration of the full PQRST, I think it's very difficult to get someone to give a picture of being a great scientist and not really being a very good scientist. So I think that as I close, I think this is my last slide. We need to also understand that we are not alone in science. There's many other stakeholders. There's the industry, there's private investors, public funders, not-for-profit funders and philanthropists, journal editors, for-profit publishers who want to make money, professional scientific societies, universities, not-for-profit research institutions, supporting staff, hospitals, insurance companies, other services, governments, state, federal, and consumers of science. And the priorities of all these people are very different. Some people want to get publishable results, scientists mostly. Others want fundable results. Some want translatable results that would work. And some want to make profitable results. So we need to find solutions that would take into account all that very complex landscape. And as a conclusion, research is the best thing that has happened to human beings, but it's very difficult, and it's also very difficult to evaluate. When it comes to specific projects, it can be almost impossible to do. And especially for truly innovative ones, I think it's just a waste of time. Research practices can be evaluated. Instead of evaluating single projects, I would evaluate the quality of the broader practices being applied. Scientists rather than projects may be easier to evaluate. Not perfectly, and some tools are better than others, but I think we can take a shot at that. Any solutions about changing research practices should enhance the science, not add bureaucracy. I think we need more empirical research on research. This is a field that is very dear to me, so I'm asking for more funding, please. Finally, the adoption of optimal research practice can be key to the success rates, efficiency, credibility, and legacy of scientific fields and of academic institutions at large. I will leave that slide with some of the unfortunate people who have worked with me and I have tortured with all of these empirical analyses that I showed you today, but I'm grateful for all the hard work that they have contributed. Thank you.